Good afternoon, my name is Matthew Raybould and I'm from the Oxford Protein Informatics Group based in the Department of Statistics at the University of Oxford in the UK. Today I'm going to present to you CoV-AbDab, the coronavirus antibody database as part of the COVID-19 special track at ISMB. And thank you very much to the organisers for selecting our work to be presented today. Neutralising antibodies and antibodies will play a central role in combating COVID-19, whether that's in better exposure diagnosis, vaccine efficiency, or therapeutic design. Many will know that the University of Oxford has been actively involved in vaccine design, and OPIG are naturally happy to help wherever we can with that. But we also have a long-standing history of designing tools to improve therapeutic design, and so we're very keen to get working on the datasets as soon as they came out for coronavirus. So the first thing we wanted to find out was how much data actually exists on coronavirus binding antibodies. And we were met with a huge deluge of preprints, especially about SARS coronavirus 2. So between January and March, 1,100 were released for SARS coronavirus 2 on bioRxiv and medRxiv. In the next 2 months, a further 3,400 were released. And so this data is growing at a huge rate and it's very hard to keep track of. There's also a large body of existing work on the former coronaviruses, SARS coronavirus 1, which caused the 2003 epidemic, and MERS coronavirus, which caused the 2012 epidemic. So building on our expertise in creating databases of antibodies, such as the CoV-AbDab structural antibody database and TheraCoV-AbDab, the therapeutic structural antibody database, we wanted to create CoV-AbDab, the coronavirus antibody database, as a central resource of coronavirus binding antibody data. To do this, we searched the scientific literature for any antibodies or nanobodies that bind to SARS, CoV-1, CoV-2 or MERS-CoV. We searched in PubMed, GenBank, all of the archives, as well as Google Patents. And what we found was that sequence information, while often not present, was actually there in many cases, but was scattered in terms of its location. So sometimes in the main manuscript, sometimes in the supporting information, sometimes scanned in to the document, which means that conventional text mining approaches wouldn't have been effective in this state. So again, this further motivated us to put it in one central location with a consistent formatting. And we believe that this will help vaccine profiling efforts and comparisons to known binders. So some reviews were particularly helpful for filling in the gaps, and I've listed those here just to give them full recognition because they were extremely useful. And if you want to try out CoV-AbDab, please visit opig.stats.ox.ac.uk/webapps/coronavirus. In terms of metadata for every entry, we of course have the published name of the antibody or nanobody. We also have antigens that the antibody or nanobody has been proven to bind or neutralize. That's with an actual experimental assay, for example, ELISA, or a pseudovirus assay for neutralization. The protein domain targeted by the antibody or nanobody. So this includes not just the protein target, but also if we can find it, the particular domain. So the receptor binding domain is a good example of that. The developmental origin of the antibody or nanobody. Sequence information, so ideally the full FE or VHH sequence, but if not, at least the CDR3 regions, as well as V and J gene germline assignments. Links to any solved structures of the antibody or nanobody, and if none exist, we've built a homology model of the antibody using our in-house homology modeler, AbodyBuilder. We have references to the primary literature so that you can go back and see the context that the antibody was found in, as well as timestamps to show when the antibody or nanobody was added and last updated in the database, and any steps we're now taking to update the entry. So as of June 27th, there are 758 entries in CoV-AbDab, 171 have been tested against MERS-CoV, 367 against SARS coronavirus 1, and 509 against SARS coronavirus 2. We have full FV variable domain sequences for over 88% of entries, and we're trying hard to trace the rest by contacting corresponding authors and by tracing preprints that haven't yet released data. Currently we have 14 of those that we're tracking. We have solved PDB structures for 33 of these antibodies, which represent about 60 PDB structures, and we're expecting another 9 too based on the preprints we're tracking. So I'll now play and talk through a pre-recording of using the CoV-AbDab database in browser, and again the URL if you're interested in checking it out. So this is the homepage of CoV-AbDab with a link to our preprint. The first thing I'll talk through is our downloads tab. So everything in CoV-AbDab is freely downloadable. So we have the CSV database file, the JSON ANIKI numberings, zipped files for PDB structures and homology models, as well as an Excel file of all the data sets we're currently tracking. So what I'll do here, first of all, is bring up everything that's in coronavirus AbDab by searching over all attributes. The search returns 758 results, and you can see the main table here, which lists all of them in turn. So we have the name, the neutralization profile, the protein and epitope targeted, as well as the origin of the sequence. If we could find it the full sequence, so VH and VL. All the germline assignments that were recorded by Anarchy, as well as the CDR3 sequences separated out to allow for immediate clonotyping, any structures, and then homology models, if there are no such structures. And you can use our in-browser molecular viewer to rotate the structure and see what it looks like in 3 dimensions. We have the source literature, the timestamps, and if we are taking any steps to update the entry. So going back now, we'll do a specific attribute search. So we're now going to look for antibodies or nanobodies that bind to coronavirus 2 and that are neutralizing against SARS-CoV-2 as well. And we're going to restrict the heavy V gene to one that we might have found from our studies as being interesting. So here, 353. And we can see that there's already been several antibodies, 31 in fact, including this B38 with a solved PDB structure that belong to this germline and neutralize SARS-CoV-2. You can also search CoV-AbDab by sequence, so either a full VH or VL sequence, or just a CDR3 sequence. So here, searching with B38's VH sequence, we get 4 matches of equal length. So obviously B38 itself, but also another antibody found called COVID-207. This was from a different study. Has 94% VH sequence identity. And we have an in-browser alignment here, where white is a sequence identity match and red is a mismatch. And you can see the H3 is sequence identical, and there are sporadic mutations throughout the VH. And you also have the separate CDR3 analysis as well. So you can see here again that the H3s are identical in these 2 antibodies, and you can look at the closest 10 that are next most identical. We'll now run quickly through a bit of database analysis. And so we looked at the developmental origins of every antibody or nanobody we found that's binding to SARS-CoV-2, MERS-CoV, or SARS-CoV-1. That's going back in time. And what we found is that more nanobodies are being isolated now that we know bind to SARS-CoV-2 than we found, considerably more than we found bind to SARS-CoV-1. There's 2 reasons for that. Nanobodies are continuing now to play a huge role in our therapeutic arsenal, but also the SciBody technology is allowing us to do nanobody phage display. We have a smaller proportion of immunized animal antibodies, which is a continuing trend that we see throughout drug discovery, and a much greater percentage of antibodies being directly baited from human serum, so skipping the phage display process. I should mention it's important that these differences are considered if we're comparing binders to the different viruses. In terms of protein targets, the receptor binding domain is an increasingly dominant target for antibodies and nanobodies, which isn't entirely surprising because it's a well-known mode of neutralization to block the reaction between the SARS-CoV-2 or MERS coronavirus receptor binding domain and the natural human receptor. And this could result from a kind of bias that we are now using RBD baits to fish binders out of serum. And there's also a much reduced proportion of uncharacterized binding sites, only 2% of binders to SARS-CoV-2 that we don't know precisely where they bind. So in terms of heavy V gene germlines, these also yielded some interesting insights. So the IGHV330 germline is overrepresented in both SARS coronaviruses, but is not represented nearly as much in MERS coronavirus. And this could be a sort of key germline in terms of cross-neutralization between SARS-CoV-1 and CoV-2. IGHV323 is overrepresented in SARS-CoV-1, but not SARS-CoV-2, which could be interesting as well. In fact, the IGHV353, which is overrepresented in SARS-CoV-2, and for which we searched the coronavirus database just a couple of minutes ago, has actually been structurally characterized now in several studies. And this looks like it's a CoV-2 unique epitope. MERS coronavirus, on the other hand, has a more classically broadly neutralizing response that's reminiscent of hemagglutinin response to flu, and it uses the IGHV169 germline extensively. So we're very keen for CoV-AbDab to become a community resource, and so if you know anyone or you personally have written a paper on molecular characterizations of coronavirus antibodies and antibodies, and we don't currently have your data in our database, please do send us your data, because we think it would be really useful to have all of this in one place. And if you have performed B-cell receptor repertoire sequencing on COVID-19 repertoires, we would also really like to hear from you if you can publicly release your data, because we'd like to add it to our observed antibody space database too. And just a reminder that all of this work is on a preprint on the bioRxiv preprint server, and this is the link you can access it with. So there's further analysis that I didn't quite have time to share with you today on that preprint. And just to acknowledge my co-authors, Alexandra Kovacic, who was particularly helpful in finding the source literature right at the start of the project, Claire Marks, who helped out extensively with creating the web application, and of course my supervisor, Charlotte Dean. I'd like to acknowledge my DPhil funders, the EPSRC, MRC, University of Oxford through the SAB CDT, and GSK, UCB, Roche and AstraZeneca for funding throughout my project. And finally, all the members of OPIG for their community spirit that's made staying productive through this pandemic much, much easier. And thank you again to the organisers for allowing me to talk today, and I'm happy to take your questions in the live question and answer session now. Thank you.